Very good morning, students. Today, let me explain you about a topic called Morphology and Life History of Obelia. So, this comes under phylum Coelenterata. So, in this first slide, you'll be observing some diagrams which are related to the structure of Obelia and the life cycle of Obelia. Introduction. Obelia is a sedentary colonial marine cnidarian or coelenterate which grows upright in a branching tree-like form and has several specialized feeding and reproductive polyps. It is commonly called sea fur and exists in both asexual sessile polypoid stage and sexual free-swimming medusoid phase. That means polyp is a asexual phase and it is sessile structure, whereas the medusoid is a sexual individual and it is a free swimming one. There are some common species of Obelia which are as follows: Obelia geniculata, which is commonly called knotted thread hydroid; Obelia longisimia, commonly known as sessile hydroid; and Obelia dichotomia, it is commonly called sea thread hydroid; Obelia bidentata, which is commonly called double-toothed hydroid. Coming to habit and habitat. Obelia is cosmopolitan in distribution, only exception being the high Arctic and Antarctic seas. That means it is absent in high Arctic and Antarctic seas. They grow in shallow water, in intertidal rock pools, and are usually found up to 80 to 100 meters of depth from the water surface. The Medusa stage of Obelia species is commonly found in coastal and offshore plankton around the world. The colonies of Obelia are often found as a delicate fur-like growth on the rocks, stones, molluscan shells, seaweeds, wooden pilings, and weirs. Obelia geniculata normally grows on kelp fronds, especially on Laminaria hyperborea in conditions of moderate wave exposure. So this is the structure of Obelia colony, where you can see a horizontal structure at the base, which is commonly called as hydrorhiza, which provides the anchorage or the attachment component of the colony. And from this hydrorhiza, you can observe the vertical structure, what is called as hydrocolus. So in Obelia colony, there will be many such hydrocoli, the plural form. And you will also observe some different types of individuals or structures here, because this Obelia, as you know, it exhibits polymorphism, and the individuals seen here are a hydranth, which is also called gastrozoid, and blastocyle style, which is also called gonozoid. And as you know that hydrozoid or hydranth, it is responsible for nutrition, whereas gonozoid is responsible for reproduction. Because in this gonozoid, you will observe the presence of many saucer-shaped projections, what are called as medusae, and there is an opening what is called as gonopore, and through which these medusae will come out at the time of reproduction. Whereas in case of hydranth or hydrozoid, you will observe an elevated portion at the tip, and that is known as hypostome, and this hypostome consisting of mouth, and that is being surrounded by a number of tentacles. And this hydranth has only the tentacles but not the gonozoid, because that is not required for gonozoid as it is involved in reproduction. And you will also observe certain features here, that the entire body of this Obelia colony, it is being externally surrounded by a layer what is called as perisorc, and This perisorc it encloses a living tissue, what is called as cenosorc or cenenchyme, and the inner layer is what is called as gastrodermis. Coming to morphology, Obelia is a very small marine hydroid. It looks like a small branching tree exhibiting whitish or brown color. The height of Obelia varies from 2 cm or more. The body of Obelia consists of two kinds of filaments, horizontal hydrorhiza and vertical hydrocolus. Hydrorhiza, as I said already, it is the root of a hydroid. Hydrorhiza is the basal part of the colony consisting of tubular processes called stolons. It encrusts over the surface of the substratum and helps in the attachment of the colony. Hydrocolus. It is the stem of a hydroid. A few small vertical filaments 2 to 3 centimeters long arise from the hydrorhiza, and these are called hydrocolli, singular hydrocolus. Each hydrocolus branches alternatively, each of which terminates into a polyp. The polyps collectively are termed as zooids, and these zooids are nutritive in function and help in feeding, and such zooids are called gastrozooids, which I already told you. The axis of proximal branches bear cylindrical reproductive zooids, which are termed as gonozoids or blastozoids or blastostyles. So this is another picture showing the details of the structures present in an Obelia colony, which thing I already explained to you. The perisorc, the outer layer, the inner layer called gastrodermis, and or which is also called cenosorc, and there will be the medusa buds, and there will be the hydranth or hydrozoids having tentacles, and there will be the gonangium, or which later transforms into gonozoid or blastostyle. Coming to the living tissue of Obelia, which is nothing but cenosorc. The whole colony of Obelia, hydrorhiza, hydrocolus, and zooids contain a living tissue called cenenchyme or cenosorc. The cenosorc is diploblastic, comprising of two layers, outer epidermis and inner gastrodermis. A middle non-cellular layer of mesoglia is present in between epidermis and gastrodermis. A narrow canal called cenosorcal canal runs through whole colony of Obelia, which is continuous with the gastrovascular cavity of the zooids. The continuity of the canal system helps to transport the digested food throughout the colony. Coming to the protective covering, that is perisorc. Entire colony of Obelia is surrounded by a protective covering called perisorc. It is non-cellular, tough, transparent, yellowish brown, and cuticular in nature and is called perisorc or periderm. It makes the vertical part of the colony firm and rigid because there are the actions of tides, and there might be the damage to the colony, and that is going to be prevented by this perisorc because of its rigid nature. Perisorc is secreted by the epidermis and is separated from the cenosorc by a thin fluid filled space. However, the cenosorc and perisorc are in contact, making the colony more rigid. At some points, the perisorc is arranged in flexible rings called annuli, and these allow the swaying movements due to the force of water currents. The perisorc of hydranth is termed as hydrotheca, and that of gonozoid is called gonotheca. Morphology of a gastrozoid. Gastrozoid of Obelia is a feeding polyp. Its function is to feed the whole colony. Gastrozoid is tubular and diploblastic zooid with a central gastrovascular cavity continuous with the cenosorcal canal. The polyp is attached to the hydrocolus by a hollow stalk, while its distal end is produced into a conical elevation called manubrium or hypostome. The apical portion of the manubrium bears a terminal mouth encircled by numerous long, solid tentacles, often 24 in number, loaded with nematoblasts. The perisorc of gastrozoid, called hydrotheca, is transparent and club-shaped, invaginated as a platform or shelf at the base of the gastrozoids for polyp to rest. As I already explained to you, these tentacles present at the free end of the hydrant or hydrozoid, they are responsible for the collection or the trapping of the prey, and once the food is taken inside, they are going to be digested inside. And these tentacles are provided with a number of cells, specialized cells called nematocysts, or what is called as stinging cells, which helps to hold the captured prey very firmly. The gastrozoid and hydrotheca collectively form hydrant. In case of any emergent situation, the polyp can withdraw itself into the hydrotheca and the tentacles fold over the manubrium, covering the mouth. The presence of shelf prevents the polyp to retract into the hydrocolus. The annuli of the perisorc present around the stalk of polyp allow the swaying movements due to the force of water current. Morphology of a gonozoid. The gonozoids, also called blastozoids or blastostyles, they are cylindrical, rod-like reproductive bodies present in the axis of hydrocolus and stalk of gastrozoids. Gonozoids are less in number than gastrozoids, as these are present only in the proximal part of the colony. It has a reduced gastrovascular cavity and is devoid of mouth and tentacles, which is not required as it is reproductive in its function. It thus cannot feed and receives food digested by the gastrozoids and transported through the GVC, that is gastrovascular cavity. Like other parts of the colony, gonozoids are also enclosed in a perisorc called gonotheca. It is constricted distally and constricted by annuli proximally. The apical part of the gonotheca has an opening called gonopore. Gonozoid produces numerous small medusae or gonophores by the asexual process of budding. Mature medusae detach from the gonozoids and escape into the surrounding water through the gonopore. The gonozoids, gonophores, and gonotheca collectively form gonangium. Morphology of a medusa. Medusa of Obelia is radially symmetrical, umbrella-like zooid which measures approximately 6 to 7 mm in diameter. The outer surface of medusa is convex and known as exumbellar surface, while the inner concave surface is called subumbellar surface. A short manubrium containing a quadrangular mouth at its distal end hangs from the center of the subumbellar surface. The medusa is craspidote type, as its edge is produced inwards into an insignificant rudimentary vellum. The margins of the medusa bear initially 16 short contractile tentacles which gradually increase in number. The mouth opens into a short gullet which leads to a wide expanded stomach from which arise four narrow radial canals which mark the four principal per radii. The radial canals extend till the margin of the umbrella and open into a circular canal running parallel to the margin. The radius bisecting two per radii is called interradius, which are 4 in number, and that bisecting per radius and adjacent interradius is termed as adradius, which are 8 in numbers. The tentacles present at the end of these radii are named accordingly, such as perradial tentacles, interradial tentacles, and so on. The whole system of canals is lined by inner layer of gastrodermis, and both the exumbellar and subumbellar surfaces are covered by epidermis. Nervous system consists of two diffused nerve nets which are concentrated around the margins of the umbrella and form two circular nerve rings. 8 receptor organs called statocysts are present at the bases of adradial tentacles, and these are the organs of balance, muscular coordination, and equilibrium. The medusa possesses 4 gonads on the subumbellar surface, and these are perradial in position, and each of these is present in the middle of each radial canal. These are dioecious, that is, sexes are separate, wherein male and female medusae being separate individuals. So here in this slide you will observe that structure of medusae. Reproduction in Obelia. The life cycle of Obelia includes both polyp and medusa stages. Polyp is an asexual form and reproduces by asexual means, while medusa is a sexual zooid and reproduces sexually. Asexual reproduction. The polyps are reproduced asexually by the process of budding. The hydrocolus gives rise to a number of gastrozooids, and as the colony matures, blastostyles bud from the axils of proximal gastrozooids and hydrocolus. Each blastostyle produces a large number of medusa buds in spring and summer. These buds gradually develop and mature. When fully formed, they detach from the blastostyles and escape into the water through the gonopore, which I already shown you in the previous slides. So this is what the detailed structure of the development of medusa buds in Obelia colony. So as you observe here, this is what gonozoid or the blastostyle, where inside which you see a number of medusa buds are there, and there is an opening called gonopore, and when these medusae are matured, they escape out of this blastostyle through this gonopore, and they are released freely into the marine water. Sexual reproduction. The sexual reproduction in Obelia takes place in the medusa stage, the male and female medusa being separate. The medusae produces ova and sperms and release them into the water where fertilization takes place. So fertilization is external. Sperms may also enter the female medusae along with the water current, and fertilization may also take place inside the body of female medusae. So sometimes it is internal also. The development of fertilized egg. The fertilized egg undergoes complete and equal cleavage, resulting in the formation of solid ball of cells called morula. It develops a central cavity, blastocele surrounded by loosely arranged blastomeres. This hollow blastula is termed as coeloblastula. Gradually, the new cells cut off from the blastomeres and start migrating in the blastocele from one end of the coeloblastula. Slowly, entire blastocele is filled with the cells, and hollow blastula converts into solid gastrula called stereogastrula by delamination. The outer surface of the embryo becomes ciliated, forming a ciliated larva, that is planula larva. It is double layered ovoid larva consisting of outer ciliated ectoderm and inner solid mass of endodermal cells. It actively swims in the water and helps in the dispersal of species. After a short period of time, the larva settles down and attaches itself to the substratum by one of its ends. The attached end forms a basal disc, while a mouth surrounded by tentacles is formed at the distal end. This sessile stage is termed as hydrilla stage as it resembles a hydra. Gradually, hydrilla undergoes asexual reproduction repeatedly and converts into an adult Obelia colony. So the details of this explanation is being depicted here in this slide, where you see the Obelia colony which is there on the left hand side. It is reproducing by means of asexual phase, where the daughter medusae are produced inside the blastostyle and the daughter branches are going to be produced by the hydrocolus. And once the medusae are released out of the blastostyle or gonozoid, where you can see there is a male and female medusa there. The male medusae produces sperms, female medusae produces ovum, and when they unite together, they produce a zygote, and that zygote starts dividing by means of cleavage, initially into two-celled, then into four-celled, and then into blastula, and that will produce a free-swimming ciliated larva called planula larva. And this planula larva, as I said earlier, it swims freely for some time and it settles down into a suitable substratum, and then it develops into a young polyp, and this young polyp will later develop into a stage called hydrilla, and that hydrilla later it transforms into an adult Obelia colony. Metagenesis. The life cycle of Obelia represents a remarkable example of alternation of generation, where the asexual and sessile phase of Obelia reproduces asexually by budding and gives rise to sexual and free swimming medusa. The medusa reproduces sexually and forms new polyps. Thus, a diploid asexual hydroid phase alternates with another diploid sexual medusoid phase. This phenomenon of alternation between two diploid phases is termed as metagenesis or alternation of generations. Coming to polymorphism, thus, the life cycle of Obelia includes three distinct types of zooids. Number one, nutritive polyps, that is hydrants, asexual reproductive polyps, that is blastostyles, sexual reproductive polyps, that is medusa. This phenomenon, where Obelia is represented by structurally and functionally different individuals, is called polymorphism. Initially, the colony of Obelia is represented by only two forms, that is, gasterozoids and blastozoids, and is called dimorphic. Later, when gonophores develop on the blastozoids by the process of budding, the colony is considered as trimorphic, represented by three kinds of zooids. So, this is all about the today's class, dear students. So let me continue in the next class. Thank you.